All right, so thank you very much first for inviting me to be giving your lecture in your course, which is extremely well organized. So congratulations for all the work that you have been doing there. And I can see some of the students, and it's really great. Hope you are enjoying your day, and then you have also a very great 2 days at Eliava. So again, for me, it's really a pleasure to be here. I'm a professor at University of Minho. I do science, and I'm a researcher also in bacteriophage biotechnology. And I've been doing research for many years and writing papers. And this is what I want to share with you about the scientific writing, and more specific, how you could write a paper. So then here we go. So when we speak about scientific writing, it's because there's a reason. And of course, there's many good reasons, but the most important reason is, if you are doing research, it's important that society knows what you are doing. It's important that you report your findings, because science is always built on others' discoveries. So you have this societal obligation to communicate what you have been doing in your research. But also, as you know, those that will follow an academic or a research career, there is a need to do publications, because it's the output of your work. This is how you can be evaluated, and yeah, this is considered an output. So it's really important that you public make public your work by doing oral presentations in scientific conferences or even posters. You can also use other ways to communicate science, which are not so standard, but the scientific blogs or data warehouses, and also ultimately the written reports or papers. And this is what I've been talking about in the next slides. So we usually use it's usually the usual approach for writing papers is the INRAM format. So what is the INRAD format? The INRAD format is a format which is a little limited, but still most of the magazines or most of the journals use, and most of the scientists use. This is a format in which INRAD comes from I, introduction, L, literature review, M for methods, R for results, and D for discussion. So how do you present your findings? You present your findings describing why you did something, how you did something in your methods, what did you achieve, then what's important in what you achieve. So what? So this is the IMRAN format. And this is more or less the structure that I will be using to present the following slides. So everything is okay now? Can you still hear me?
Yes, we can hear.
Great. Then one paragraph here, just to stop. It's very important to be aware about the ethics in science. And I'm sure you also know that, but it's really very important, because many papers have been retracted due to science misconduct. And what is science misconduct? Is when the data is fabricated, falsified, there is plagiarisms or other practice that really deviate from those common accepted within the scientific community for proposing, conducting, or reporting research. So this has been punished. There is even cases of fabricated scientific results, in which even led to the prison of those who made this fabrication. So it's very important that you only report, of course, what you find, and use your own words to describe it, and use your own work. Of course, it does not include honest errors, because maybe you are not doing the right interpretation, but it's your interpretation of your data. Or maybe at the end of you did some error in your methods, and then you report it. So it's not a misconducting science. But if you find that, and if you find that for somehow there was a mistake in your paper, you should really acknowledge that, retract the paper, rephrase the paper, or even publish an amendment of this result. But this is not considered a misconducting science. Okay, now let's go back to how you write a paper, how you write a manuscript. And I have a question for you. When you start writing your writing, where do you start with? Do you start by writing the introduction? You start by writing the material methods? You start by the results? You start by the title? So can you please tell me that by doing, you see this, this is a Slido. I'm not sure if you are aware of Slido. If you can just use this QR code, or you can also go to Slido, slido.com. So just Google Slido and insert this code 891152. And then you can, this is anonymous, so don't worry, you can start voting. So you can already register. I already see one vote and one participant. Yes, thank you. You are, I will show what you did at the end. So everyone got the QR code, right? So I will stop sharing so that we can see what you are voting. Okay. This is the one.
Sorry, could we get the number again, the code for joining the Slido meeting?
Yes, sure. I will in a moment. This is the code 891152. Oh no, I'm showing you. Don't yeah, just don't follow what the others are doing because I'm showing already the results. Okay, so everyone did that?
Yes.
Yes. So let's now stop. Okay. Oh my God, I should be able to go back. All right, so I'm sorry, I cannot go back. Let me go back to my presentation. So what did you vote? Most of you said 54% at the title. So you would start by the title, and then 21% by the introduction and 21% by material and methods. So the majority of you would start by the title. So you think that you would write an article starting by the title. So let's see if you are right. Well, there's other strategies, but I'm going to show you another strategy. For us, or for me, the title is the last one to write. So you only write the title at the end when you have your manuscript done, so that you know exactly what should be the right title for your manuscript. So it's not the way you think it is, it's a little bit different. So let me tell you what are our ideas about what's the best tip and what's the best way to write a scientific paper. Okay, so you should actually, following this INVRA method, you should actually start by the results. You could not imagine why you should start by the results. Because maybe you have going to the lab, you generated a lot of data, and then you need to make good sense of your results. So you need to look at your results. You need to look at the data that you obtain. You need to see how connected are these data and what are the major findings of your article. Sometimes you want to study something and then your data will show you another thing, and then you explore that other thing, and that at the end of the story, then you have a different story that you wanted original to talk or to tell to others. So you look at your results and try to make sense of that. So after all, you know exactly what message, what novelty, how you want to explain your achievements. And then you go to the introduction, and then you do the methods aligned with the results. So introduction and methods can actually be done at the same time. And then you do discussion. And only at the end of the manuscript, you do the summary and you create the title. Okay, so the title will come last. All right, so let's now see how you start with the results, and again, why you start with the results. Okay, and sometimes I have some issues here. I need to go to the presentation again and start sharing. Sorry for that. Online not always works so well. Okay, let's see if it goes. All right, so how you start with your results. So you have your results. You can print it, you can have in a screen, wherever you find it useful. I find it very useful if you print it and then you see all your results in front of you. And then you try to make a logical connection between the tables and the figures. Sometimes you need to present the results some other way, but you still need to look at them and find what makes sense with all of your results. And then you need to prepare, well, the figures and the tables that you want to show, okay? So you need to make a logical connection between figures and data. It's very important that you don't repeat figures and tables. Sometimes we see in manuscripts that there is a table about something and there's the same figure about the same data. No, it's not what you do. It's not the best practice. You either represent in the figure or you represent in the table. Legends are really very important when you prepare your results. So the legends needs to be self-explaining, and then yeah, I'm going to tell you about this in a more detail. And then when you structure your results section, you are like telling a story to the others. So I did this experiment to obtain this data, and then with this data, I found out that this could be related to something else. And then I did this extra experiment just to explain why this data was obtained, and I obtained this. So you make a story out of your results. It doesn't need to be the chronological way how you did your experiments, but it needs to be logical in a way you are telling your data or your results to the others. So make a correct connection between all your results and tell this story. We see here an example about results in which you describe what happened. For example, in this paper, we saw that temperature and shear shake were changing the size and the morphology of the cells. And then we wanted to see how phages infect these different cells. So we were explaining this, what we did and what was the data we obtained. If you have statistics related to this data, you should present in your results sections. And you also say that you obtained this according to table 1 or explain in figure 2 or everything. So you have everything explaining your results. It's not an interpretation of the data, but it's an explanation of the data. I obtained this was the statistic, this is relevant, that is not relevant, those are the results. And with these results, I did a further experiment, which I obtained this or that results. This is like a story of all your results that you are explaining here. Okay. So, not sure why this does not go further. Oh, sorry, it was too fast. Yeah, there is some delay with the Zoom, so this is why I'm having some troubles here. Okay, so this is describe the data, do the statistical analysis of your data, and then explain what you did further. This is the story that you tell in your results section. Now about the tables. How do you present your tables? So your tables, they need to have a title, and the title needs to be very explicative of what you want to show in your table. So it's important that you give all the explanations in the table, and that the reader can look at the table without, well, it can go to the material methods, but without the need to know what did you do, just to have your data there, or in order to obtain these results. So all tables must be self-explanatory, and then avoid repetitions. And it's also important that in your tables, you display all the statistics related to your work, the variability, the variance, and the significance of the data displayed in your figure or in your table. Let's look at the data or some results here. Now you have a title, and the title really says what you have here. It's the influence of temperature and the agitation speed on the cell shape. And then it also explains what you have in each column of your table. And then you have for each column, you have units, usually what it represents, and then you have the units, right? So you also put your standard deviation if it's important. Sometimes you can also use the footnotes of the table to explain further what you want to say. For example, here, standard deviations are is exemplified here and not on the title. You can also use the footnote to explain more. Some journals, they ask that. You can also use the footnote of the table to explain a little further what you did, not be exhaustive in your methods, but what you did to obtain these results, so that you don't need to go to material methods to see what is really in your table. But it's optional, right? The most important thing is have a very informative title in which you really explain what you have, and then you have all data with dimensions and with standard deviations in your columns. All right. So now, what about the figures? So in your paper, you want to put tables, but maybe you also put figures. And figures are very important because they give some visual perception of your data. And this is very good to communicate science. It's easy to see in a figure what you want to show rather than in the table. Tables are important, but the figure is the most effective way to communicate. So it's very important whenever you can make figures, just try to make figures. Bark figures, just regressions, what type of figures that, or even drawings that makes a better way to visualize your results. But the most important in the figure is that it needs to have the name and the legend of the figure. And also the legend, as well as in the table, the legend must be self-explanatory. So you can also use different symbols to distinguish from each other in a logical way. For example, you have a figure in which you combine 4 or 5 figures, but then you have figure A, figure B, figure C, and then you put in your legend what are the figure A, figure B, or figure C. So the size of the figure is also very important. So it needs to be large enough so that you can really see the data. Sometimes authors, because of saving space, they make figures very small, and then you cannot see very well what is there. So you need to make it in a way that the reader can really see what you want to show. And also be very careful when you present data. Use bars for standard deviations. Be aware and careful with a logarithmic scale. So sometimes if you have logarithmic data, you can use logarithmic scales, but you need to use it well. This is mathematics, but you need to do a very accurate table. I've been seeing logarithmic scale tables or figures without being a logarithmic tendency of your data. So it's wrong. So you need to be very careful in the way you present your figure. Okay, this is an example in which you have the legend with, again, self-explanatory, in which you explain exactly what you have in your figure. So one-step growth curve experiments, this you know, we'll be doing that at the temperature that was been done, which is important, with the strain that was been done, and which you are also displaying the error bars so that you can see if there is any statistical relation between the two experiments that you developed. So one is a 30 degrees, the other is a 25, and there is some data where you don't have statistics significant. So it's important that you also put statistics in your figure. So this is again the same type of figure, and this is an electrophoresis, for example, in which you really need to say what you have, what's the ladder, what is here displaying. So everything needs to be well explained. And now I'm going to, so this is a logarithmic scale that I was telling you about, and this is all the details that you have to give to your figure. Okay, be also careful with the units that you display in the figure. Okay, now we have a figure which there's something wrong here, or maybe more than one thing wrong. So would you mind to go again to Slido? Now shift this here. And just to the QR code as you did before, and I'll be more careful now not to show the data. And please, I will soon activate that. Start writing. This is what you see wrong in this figure. Okay, can you please start and writing down? Is it possible to write?
No, because QR code is not full.
Can you use this 1433626? Okay, I can see that you are already writing. Very well. I can see already 3 votes or 3 interactions. Perfect. Look at the figures, see what's wrong. Can make short answers. Nadia, can you still hear me?
Yes, Joanna, we can hear.
Because I'm connecting another screen and sometimes it's it doesn't work so well. That's why I'm asking.
Yeah,
you.
we can hear.
Thank you.
If there would be a problem, we will also
You
inform
tell
you
me.
from our side.
Okay, great. So there is still people typing. I will wait for a while. We have 11 interactions already. You can still see the figure, right?
Yes, we can see the figure.
Wait for a while because I see that still people typing. I will only show you when everyone is not to influence your opinion. Great. I have 16 opinions already, 2 waiting. Thank you very much for your collaboration. It makes it more fun. Okay, just finalizing. Only one typing, and yeah, I think that I can. So who is finishing? I will wait for a while. Okay, maybe I can already start showing some of what you've been telling me. What it's... Let's see if you can see. What is wrong in your figure? It's show between 0 and 6 hours with 100 micrometers, both of them looking similar, as there is no effect at all. Realistically speaking, the timing means 24 or 12 hours should be less than predicted. I'm not sure whether I understand your answer, but the thing is that there is no connection between what's in the figure and what is being described here after 6, 12, and 24 hours with and without. So we don't know what is what. So we don't know what is this relation to the time. And I think this was what you intended to say here. Very well. And there is no title. Yeah, the figure has no title. It doesn't mean that you are obliged to put a title here in the figure. If you write a legend, you really actually don't need a title. So there is no title, there is a wrong title, something that we even don't understand what's the title. But if you have a legend, you don't even need to write this title. Legends are really very important. Yeah, the titles of the graphs are missing in the figure. Of course, you don't know what this is, what is this. You just know it's 100 micromoles. Probably it's about XTT, or the farnesol that was used, but we really don't know what's this color. So there is no legend that is explaining what's going on here. So again, no self-explanatory legend. Very well. This is wrong. Also, and very importantly, you plot it. There is no title in the X in the in the X is legend. So there is nothing here. There is no legend. So we don't know what's 0, 0 0.2, 0 0.5, what it is, concentrations, absorbance, intensity of fluorescence. So we have no scale. We don't know what it is. And we don't know if 2 is what? Fluorescence intensity, if it's nanometers, if it's number of cells. So there's nothing. So this table doesn't tell us anything. Okay, great. This is also wrong. And also, yeah, the title, the self-explanatory is not enough. We've seen that. This is a very short legend without explaining what's really is in the figure. Again, the X without the name. The data isn't very distinguishable, and the title of the axis isn't there. Perfect. We've been speaking about the title of the axis, right? But also these are so fade colors. So when you want to represent different data, you also need to show differences between what you are showing. So this is all pink and very faint, so printed, you can hardly distinguish this. And if you had a legend on this, you can also not distinguish that very well. So very well, you are really picking the most important things here. Axis definition again, title not descriptive and not self-explanatory, and the symbols are too small. It's also true. We don't see it very well. The symbols are very small. Also, the X definition is not there. Bar charts. Okay, bar charts. Yeah, bar charts are sometimes not so easy to visualize, but it's okay that you can use bar charts, but you need to be careful when you use it, and you need to use it for only certain types of data. It's more detailed. The y-axis is brushing the date of 300, so I did not notice that. But yeah, there is something wrong here in the 300 micrometers. You notice that they are all equal, so there is no distinguish between that. And then if you have standard deviations, you can actually say what is statistically different in your figure, and this is not written here. And again, the title. Okay, so I think that you really mentioned the most important aspects of what is wrong in your figure, or what is wrong in this figure. So again, use sharp and different colors to distinguish your data. If you use bar figures, use it in a correct way. Don't forget the legends of the axis. And this one has no legend, and units. Don't forget the units, and be very careful with the legend of your figure. It needs really to explain exactly what you obtained. Okay, thank you very much for your suggestions. Very good, you did it well. And let's skip now or start talking about the introduction. So we remember, we are writing an article and we started by the results, okay? We make sense of all the data you obtain. You see how we can structure the results section so that you can tell a story with your results, and then you work in your figures and in your table just to present them in the correct way. And then you have your results sections written. Now we can start with the other parts of the article. So then you can start with introduction. And how would you structure your introduction? Why you start now with your introduction and not before? Because maybe, as I said, explain in the beginning, maybe you wanted to obtain some data, and then at the end you obtain some results that led you to some other experiments that led you to some other results. So this is when you see them and when you make the story of your results, then you know exactly what information you want to tell to the others, to your scientific community. And then you will only start doing the introduction when you really know what you want to tell, okay? And then you start by the introduction. So usually you use first a few sentences that will introduce you into the area of the research. What's the scope of your work? Is it about phages? Then you can say phage therapy has been very important and has been saving many lives. You can start by something like that, very general, just to bring the reader to the context of your work. And then you describe the state of the art. So what is known about this topic? What is important that you say about this topic? So that it will lead you to the existing gap, what it does not know or what you really want to show. So then you explain the gap of knowledge that comes from the state of the art. And then it leads you the way to explain what you aim, what you wanted to do, just to fill the gap that you explained before. So this way you make the introduction very interesting to the reader. So first you just focus his attention to the scope of your work, you describe what is known, then you say, okay, everything is known, but there is this part that still needs further insights. So this is why my work did this, I did this or that. to fill in this gap of knowledge by explaining what was your aim. In some of the journals, you can already say something about what you did or the major findings of your work in the introduction, but this is not compulsory. What you really need to focus very well is on the aim of the work. And the aim of the work needs to be clear, because this is this aim, with this aim, that justifies the experiments that you developed, the results that you obtain. And when you do your discussion and your conclusion, you need always to address this aim. So you cannot say, okay, I am aim at characterizing this phage morphology, and then at the end you conclude something about the phage physiology, for example. So everything needs to be aligned with the aim of the work. Okay, let's see here some examples of some introductions of some articles here. Introduce the topic. So it's just a general introduction to the topic, just to allow the reader to know what's the topic of your research, very generally. And then explain what you know about the topic. So what is very known about, this is the mechanisms of material defense, what is important. Again, it's the importance of the cell morphology and how phages interact with the cell morphology. So what is known about this topic, and then where's the gap of knowledge? You can always say, however, nevertheless, the influence of the infection in this different morphology of cells on progeny phage production remains unclear. So this is not known. So this is why you did this manuscript. So this is why you did what you did in your experiments. All right. So general context of the work, very general, then explain the state of the art, and then explain the gap of knowledge so that it justifies what you did in your research. And then at the end, and very important, because there is this gap of knowledge, you put the aim of your work. So this study aims at understanding the impact of cell morphology on the level of the outer membrane proteins and lipopolysaccharides that will then influence the phage interaction with the cells. So then you explain the aim of the work. Okay, so this is how you structure your introduction. Then you can write material methods. It can be at the same time, but only after the results. So because you are only had the methods and the materials that are connected to the results that you are presenting. So you need to explain what you did. You usually do a logical explanation, can be a chronological explanation. You only explain what you are really showing. So if you have data that you are not showing, you don't need to explain how or why you did it and how you did it. And very important, in your biological, if you have biological materials, you need to say what is the origin of your samples. For example, if you are using clinical samples, it's important to say, all reviewers, they ask where these clinical samples are coming from. If you are using a collection strains, you need to put the codes of the collection strains. You need to cite these collections that you where you have to retrieve your biological samples. And if you want to deposit these biological materials, or even if you are generating genome sequences, you need to deposit it in the public banks, and you need to have the accession numbers of these depositions. So most journals, or all the journals, they demand that you write the accession numbers. You can write it here, you can write also in the results, but also it's always in the material methods that you write this information. It's also important that you describe how your samples were stored, pre-treated, and preserved. So another important thing in your material methods is if you use reagents and equipment, usually it needs to be indicated what is the manufacturer of your reagents and your equipment, because it's important. For example, maybe you have also this experience when you are using culture medium for growing your bacteria, mostly the undefined culture medium. So the brands and the manufacturers, they have different hydrolysates, for example. So your growth curve vary with the type of reagent that you are using and with the brand that you are using. So it's really important that you give these names so that others can repeat your experiments and can know why you obtain this growth data, for example, because some other author can obtain with a different brand a different growth curve. And then we'll try to compare your results and we'll say, okay, so my bacteria, it growth in a higher concentration, but I was using another reagent, or I was using another culture medium from another brand. So it's very important that you also say what brand you are using. And then here are some examples in which you say the organisms that you use. For example, this is related to yeast, Candida glabrata, and a clinical strain, where it came from. And if you are using collection strains, you need also to cite the culture collection reference. It's very important. And again, the reference of the equipment. For example, here there was used some skin, three-dimensional skin. So it was referred here where the skin was purchased. And the equipment that was used to do some experiments, it's also important to write down here. Okay, reference of materials and equipments, reference of all your biological material, where they are coming from, and the collection and the cultural collection reference. So we so far did the results, how you present tables and figures. We spoke about introduction, how you structure your introduction, and then comes the discussion, which is usually the most difficult part to do, because when you do discussion of your results, you really need to read a lot. You really need to relate your data with others, what others have been finding. You need to highlight what is important, what's the most important aspect that you found, and compare that with others. So it's really important that not only for the discussion, but for all your research, that you keep updated in the literature. So you need to be always reading new papers, new articles, new blogs, new science blogs, so that you are very, very well updated in the science that you are developed. So the discussion is really comprised in four main aspects, or four main parts. The first part is what your paper, what your data contains of novelty. And this is what you first write, and you need to be aware of what's novel in your data. So you are only doing a paper or communicating science when you want to show some novelty. So it's important that you know what's the novelty, and then combine your results with the literature data. So what's novel, why it's novel, and why it's novel compared to the others' data. And if you are explaining a phenomena, because it's also depend on what type of article you are or article you are writing, if you want to explain why you obtain this data, why this is novel, then here you present a theory that can explain your results. And then be careful, because you are presenting your theory. So sometimes you need to use the word I hypothesize, this is probably this, oh, it's most likely that, so that it can give you some a way of, it's your explanation of the data. You are not saying that this is exactly this. You are just withdrawing some hypotheses. So it's very important that when you write, and this is a hypothesis that you are withdrawing, you just make clear that is your hypothesis, and don't be too sharp in explaining what you obtained. And at the end, important is so what? So You did something new, we discovered something, you combine your data with literature, you present a theory explaining why you obtained this data, and then you also finalize your article by saying why it's important. So what's the importance of my data? How can this be further exploited? How can this be good to humanity? So this is, so what part is very important in your discussion. So let me give you a brief example. So I'm bringing this example because by images you can see. So this is a work that was published some time ago, in which this work shows that there are two types of yeasts. They are in yeasts that cause diseases. This is round-shaped yeasts, is Candida glabrata, and these red or green yeasts are Candida albicans. And it's known that Candida glabrata cannot penetrate into tissues because they don't produce the IFA, so they cannot penetrate into tissues. But when this Candida glabrata is combined with Candida albicans, it can hitchhike in this IFA of the Candida albicans and can penetrate within the tissue. So this was the novelty that we found in this article. It was already known that Candida albicans, through this IFA that forms, can penetrate the tissue. So this is a tissue, can penetrate the tissue and cause serious damages. It was known that Candida glabrata, which has not this ability to form the IFA, cannot penetrate the tissue, does not create major damage. But when they are both combined, they create a huge damage to the tissue because they both can penetrate, produce enzymes, and damage the tissue. So this was the novelty of the work. So how did we construct the discussion? First of all, say what's novel. And the novelty here is that Candida glabrata can really infect the tissue when it's combined with Candida albicans because it hitchhikes, it attaches to the IFA and then can go and penetrate the tissue. So this, no one has ever observed that, and this is the novelty of the work. And then you relate this novelty with others, what others have been explaining. So it's already known that Candida albicans can penetrate, but there is not known about the Candida glabrata. So this will be related to what's been written in literature. And then you try to explain, present a theory to explain why your results. So why Candida glabrata can invade tissues when in presence with Candida albicans. And then you will present here a theory that can explain your data. And then at the end, so what? How these, all my results can lead to anything impactful. And then here, at the end of your discussion, you say the importance of recognizing the occurrence of such mixed infection or combined infection is important because it really highlights the damage that co-infections causes, which is very important when you are trying to diagnose infections caused by Candida albicans. So then at the end, you explain why your data is important, and now you can further explain your data. So this is how you structure your discussion. Okay, I need to move a little further because it's 10 minutes before the end of this talk. Let's now go to the summary. So when everything is almost ready, you are delighted because you just need to write the summary. And the summary is easy to write, but it needs to be a very appealing summary. You need to, in your summary, try to explain everything that you made in your article, but in a very appealing way. So usually you start by the scope of the work. So what's the scope? What's the area of your work? What's the aim? The aim is already here. You remember the aim was in the introduction. The aim is here in the summary again. And what you did to accomplish your aim. So a brief introduction of the methods that you used to accomplish your aim. And then what were the major results, and then the conclusions. So this is why and how you structure your summary. You need to move further. So there's another different way you can present it. Some journals you can just present as a whole text with this structure. In other journals, they already want you to make parts of your summary in which you already present the background, the methods, the results, and the conclusion. So these vary in different sections. But the alignment is the same. Again, the theory, how you develop the aim of the work, and how you develop your experiments to answer to this aim, the main results, and what you conclude from your work. And after that, you reach to the title that you wanted to start first when I asked you where you would start, and you would start with the title. Now see, the title comes at the end. When you have your article finished, then you want to see, then you know exactly what you are trying to tell to the audience. So then you are in a better position to write the best title for your work. And the title needs to reflect the content of your article, and it's important to be short. You cannot put very long titles. It's not good. It needs to be short, but it also needs to be appealing. And then you can try to omit some redundant phrases such as a study of, a report of. It doesn't need to be done like that. So for example, this is a title, which is a good title. Assessment of soil erosion in Africa using remote sensing. It's informative, it says what the article is about, and it's also appealing. So it has the subject, assessment of the soil erosion, so it tells you what you did, it tells where you did it, the location, and it also tells you the method you use to in all your manuscripts. So you use the remote sensing. So it's a very informative title, a short title, and an appealing title. Okay, let me ask you for another challenging now here is for you to select again in Slido, for you to select, and here you need to be fast to select what from these titles that you see here, 1, 2, 3, 4, and 5 are the best to describe a work, an hypothetical work. Okay, let me activate this one. Okay, you can already start voting. What do you think is the best title? The first one. Can you already start?
I think it doesn't work, the QR code.
Okay.
I mean, it works, but there is no active poll. It says when we scan it, it says no, there is no active poll.
Okay. But I have one voting. No one was able to do that. No. Okay, but you can ask there. So you can raise your hands. This is another way to do it. So who thinks that the title number 1 is the best? Development and application of a genetically engineering bacteriophage carrying CRISPR-Cas components designed to specifically target and disrupt the horizontal transfer of multiple antibiotic-resistant plasmids. Who think this is a good title?
No one.
No one.
No one.
And I can see you. 2. CRISPR-Cas bacteriophages suppresses horizontal transfer of antibiotic-resistant genes in E. coli. Okay, E. coli is not italicized, but I'm just speaking about the content, not the format. So who thinks this could be a good title? Some hands, many hands. Great, good. Designer phages against resistance. Who thinks this is a good title? No one. A study of how gene editing in phage can stop resistance from spreading in bacteria. A good title? No one's. And this one, supercharged viruses that fight back. No. All right, I think that you catch the idea with these very strange titles. Of course, the first one was too long. Too long, you don't need this title. It's really long. This is the best one, and you really pointed well. This one, Designer Phage Against Resistance, is too vague. We don't know what this is about. If you read this title, you don't know what the authors did, what was the study about. A study of how gene editing in phages can stop resistance. It's better, but it's still not very specific. This one is specific. This one, you say what was the method you used to suppress horizontal transfer of antibiotic resistant genes. And it also did explain in which bacteria was used. And this one is not scientific at all. It's a very informal and not scientific title. Okay, well done. Very well. So we are almost reaching the end of this session. And this is a table of a very useful table in which what you should do and what you should not do when you are writing an article. So I've been telling you some of these do's and don'ts throughout my presentation. So then you can also have access to these slides. So you can have a look at this. And when you do your work and when you start to do your work, just carefully look at these do's and don'ts and be sure that you do the do's and don't do the don'ts. So it's important to just go through all these. This is very useful to see and to check that you have a very nice publication. So my final slides is about artificial intelligence. And why do I'm writing and speaking about that? Because we have access to several platforms of artificial intelligence that can be very helpful in writing an article. But they will not write your article. They are just a tool to help you writing the article, but they don't replace you at all. Otherwise, they would not need, or we would not need science or scientists. And I've been asking ChatGPT about this. Can I use AI to write a scientific paper? So AI gave me a very bright and interesting answer. Yes, it can be done. You can just use chat just to make some brainstorming, just to help you generating your hypotheses, maybe suggesting structures of your article, but it's not going to write it for you. It can also help by editing your texts. You can write, if you have some difficulties in English, you can use artificial intelligence to correct grammar, to correct your tests. But you need to be very careful because sometimes it does not understand exactly what you are saying and will change the order or the idea you wanted to show. And it can also help you construct your data. But you need to be very careful with artificial intelligence. So you need to be careful because sometimes it does not produce and does not give accurate results and accurate data. Sometimes it does not help you to do originality or help you to have originality in your work. And also there is an ethical concern because you cannot cite artificial intelligence, and some of the articles, they don't allow you to use it, or they allow you to use it, but you need to disclose it very well, how and why you did it. And you should be aware that you need to have critical thinking, and you cannot use AI to replace your critical thinking. So we need to be very careful with AI, and we need to we use it as long as we use it as a good practice. And the best practices or the good practices in using AI is always verify sources and citations, because AI just invents citations, and you can try that, and it really cites things that doesn't really exist. So you need to be very, very careful. You need to disclose the use of AI to the journals that require, and most of the journals they require. And you can use AI just to assist you, just to help you. The best to use it is to correct your grammar, to correct your texts, texts, but even though you need to read it again and see if it makes sense. It will never, never replace you. And keep your critical thinking. Don't use AI to make the interpretation of your data because it can create bias, because it will use others to interpret your own work, and it will create wrong interpretation and will introduce bias. So it can be dangerous. So you need to use your critical thinking. So my advice to you to use AI is just forget about it, just write your article, and then at the end, just use it for improving your writings, just to improve your writings or to improve your figures, not to replace at all your work. And with that, I finish wishing you all good luck for everything in your life and also for your writing.